Yes, it's here.
All right, should I go ahead and start? I think I'll go ahead and start. Did somebody have a question?
That was just me hitting the wrong emoticon, sorry.
Oh, I see. Okay, that's what that was. All right. Yeah, we're getting a lot of thumbs up to get going, so yeah.
Great.
Go ahead.
Great. Okay, so the paper that we're doing today is this one, binary and analog variations of synapses between cortical pyramidal neurons by Sven Dorkenwald and a whole bunch of people at the Allen Institute. This paper is based on this preliminary data set that the Allen Institute took using regular TEM serial sectioning collection on TEM grids that they did actually as part of the phase 1 of the Microns project. And the data set looks like this. It was a hard-won data set. It's got a lot of issues, and it does make me wonder. Their more recent data sets have much larger volumes, many more cells, and I personally do not know if they have redone this type of synapse study in those newer data sets. If they've done it with different types of neurons. This paper does layer 2-3 pyramidal cells connecting to other layer 2-3 pyramidal cells in this volume. But this paper has way more statistics than any other paper that I've seen that looks at these types of matched synapses. So I'll go into that in a second. In any case, this is a 140 micron by 250 micron by 90 micron thick connectomic reconstruction of the layer 2-3 area of the visual cortex, primary visual cortex of a mouse. The sectioning was at 40 nanometers thick. This image from figure 2 is actually showing all of the cell body locations within this volume. So they're all of the, I think this is just showing the pyramidal cells. They also talk a bit about connections to inhibitory cells and other types of cells. But the main analysis is from layer 2-3 pyramidal cells to layer 2-3 pyramidal cells. And so all of the dots here are 362 layer 2-3 pyramidal cells where the cell bodies are. The gray lines connecting them are single synapse connections. So there was a single synapse between those two cells. The blue lines are ones where there was two synapses between those two. And what do I mean by that? This is an example of this. So here is two pyramidal cells. They trace the axon of one of those pyramidal cells. I should know the. They trace the axon so that they can see where it's synapsed over here on the gray cell, and it's synapsed in two different places here. It's synapsed on at this location here. I hope everybody can see my mouse, and at this location way over here on two different dendritic branches. But the key point is that both of these synaptic connections have the same presynaptic cell and the same postsynaptic cell. And that is crucially important to this paper because the idea is that these matched synapses, these synapses that have the same pre and postsynaptic cell, should also have the same learning history. They should have the same local learning history as each other. And therefore, if there's something like Hebbian plasticity, they should be correlated in size. They would both learn to strengthen at the same time, or both learn to weaken at the same time if it was a LTD, a long-term depression scenario. They've got a ton of these cells. They've got a lot of these blue connections that that are double synapses that they can look at the statistics of. And they've even got a whole bunch of these red connections, and the red connections are when two cells share more than two connections, so 3, 4, or 5 connections they saw. Now, go ahead and stop me if anybody has questions, by the way, and somebody let me know if somebody has raised their hand or something. Okay, Randall.
Yeah, that sounds good. I can tell you if someone raises their hand.
Okay, great. Okay.
We have a raised hand right now.
Go for it.
Sinem is asking a question.
Go for it. Yes?
Hello, Sinem. Can you speak? Or do you want to put your question in the chat if your microphone isn't working?
Anything?
What about now? Can you guys hear me now?
Yes.
There it
Okay,
goes.
perfect. So these singlet, paired, and quadruple synapses, are those just the ones that they've analyzed, or are those like really we only found like 2 synaptic connections in these 2 cells? Because I thought that there were supposed to, like, these horizontal connections are supposed to have a lot more connections with one another, and even in the picture that you showed, there was a whole nother axon that had, that looked like it was like coming very close. Yeah, two other exons.
They
3.
they make a point to stress that this is that there might be more synapses here that they haven't counted for various reasons. One is that if the axons go outside of the volume and then come back in, there's no way for them to know that it's the same axon. So that could be a significant number. They are almost certainly have problems in the data set where they cut off axons, and that might have lost some synapses. So it is definitely the case that this is not a comprehensive, although I got the impression from the paper that they tried to be as comprehensive as possible on these 300 and some odd neurons.
But they did mention that this, basically the result is kind of a lower bound. They may have undercounted rather than overcounted. Yeah.
Yes.
Okay. Thank you so much.
So they give the statistics of these connections. So 89% had only one synapse between them. When I say between them, by the way, I'm talking about like from cell A to cell B. So any of all the ones that were connected at all, 89% had one synapse, 9% had 2 synapses, and then it went down significantly after that. But still, a significant number had 3 synapses and more. They give this plot here, which has the gray curve is showing the cumulative amount of synapses, or this is showing the cumulative number of synapses between 2 cells. And for pyramidal cells, it basically goes up to at most 5 in their data set. But if they look at pyramidal cells to inhibitory cells, or inhibitory cells to pyramidal cells, or inhibitory cells between each other, there's actually larger number of synapses, multiple synapses that they see. But again, they don't really analyze that. Figure 2, I don't know if anybody else caught this, but there was a problem with their labeling in figure 2 that gave me a little bit of confusion. I think these are the proper labels for their figure caption. C here is the frequency of is a histogram of the synapse sizes plotted in a log plot. So synapse sizes are in actually, now I'm confused. These used to be in minus in fractions of a cubic micron. I forget what units these are. I'm going to switch over to the paper for a second, see what units these are. Voxels. Those are voxels. Okay. So had it in my diagram anyway. Okay, so these are the number of voxels. We already know that a voxel size is 40 nanometers by 4 nanometers by 4 nanometers. The key point here is that the gray is the histogram of the pyramidal cells to pyramidal cells. And if we look at that, there is a skewing of that histogram so that there's more smaller ones than there are larger ones. Again, the inhibitory synapses are plotted here as well, but they don't really go into that. I want to point out figure F here, which is also interesting. So this is the distribution of Euclidean distances between synapse pairs of dual connections. And so if we had a case where two synapses are very close to each other on the exact same, it's like the same axonal branch and the same dendritic branch, which I see a lot, then there should be just a few microns between those. But you can see there's quite a tail here where you've got double, where these two axons are quite separated, like the one that I showed up here. So they've got a lot of statistics both on very close synapse pairs and very far synapse pairs that have the same pre- and postsynaptic cells. And they'll make use of that later. This is a rendering of all the synapse pairs that are same pre- and postsynaptic that they analyze here. And just looking at this, this is in Figure 2 Supplement 1. It's online. And just looking at this, it's obvious that there's a gigantic correlation between these. And other people had seen that before, but this is, again, more matched synapse pairs than anybody else has seen before this paper. So we can already see that there's a correlation. If we look at Figure 3 now, they show the histogram of spine volume counts versus the counts of particular spines in cubic microns now. There is a long tail. This is what it looks like if it's plotted in linear on the x-axis. So the spine head volume is linear. But it is well known in the literature that spine head volumes follow a roughly log linear log normal distribution. So it looks like a normal distribution roughly if you plot the spine head volume on the in log at the bottom. So this is for all 1,960 recurrent synapses, including just the single synapses. And already this, the black here is the actual histogram. The blue is the best fitting regular log normal fit, and it doesn't fit very well. And already they can see that a bimodal fit would seem to fit better. There's a little bit of a ledge here on the upper edge. And again, this is for all of the synapses, not just the ones that are pairs. Now, when they go over and they look at, this is the same plot I was showing over there. When they go over and instead of looking at all of the connections, including the single connections, if they only look at the pairs, the 160 pairs of synapses that have the same pre and postsynaptic cells, then it looks much more bimodal. And we can ask, why would it look much more bimodal? It looks much more bimodal because, this is what they were suggesting in the paper, that if you have two synapses between cells, then it's much more likely that those synapses will be large. And so we have essentially cut off a lot of the smaller synapses here and are just showing things that have two synapses between these two cells. And so the bump with the larger synapses seems to be popping out. Okay. So they looked at that bump and they said, the fact that this is bimodal might mean that this is actually a switch that is being turned on that says, okay, this is a strong synapse, or if it's off, it's a weak synapse. And so they modeled this with a with this type of model. They say that there are two states that the synapse can have. So there's a binary latent variable called H, and this variable H can either be small, so it's a small synapse, or a large synapse. But then there is this other variable V that is a noisy analog variable that is essentially noise on top of it, and that amount of V might be different from small synapses to large synapses. Okay, and so that's what they're really modeling here. They say, let me read this here. So E is showing the dual connections between layer 2-3 pyramidal cells. In this case, each is being summarized by the geometric mean of the two spine volumes. So they're taking these two synapses, calculating the geometric mean, and then they're modeling it by, so that's the black histogram curve back here, so mean spine head volume in cubic microns. They're modeling this by a mixture of two log normal distributions, that's the red dashed, and the mixture of these two normal distributions as a probabilistic latent variable model. Each synapse is described by a latent variable H that takes on values small and large, according to a toss of a biased coin, or of a biased coin, yes. And the spine volume, V, is drawn from a log normal distribution with mean and variance determined by the latent state, meaning that it could be bigger or smaller for small and large. The curve shown here represents the best fit to the data in D, which is this over here, which is just spine volume as opposed to the mean spine head. So they give a, I guess I deleted that. I should. They give a fit goodness They give a fit goodness there at some point that might be relevant, but it's not relevant for me because I couldn't really compare it to anything. But in any case, this binary model seems to fit the data reasonably well. They also take a look at, in G, they do a comparison of the spine volumes for single and dual connections. So the single connections, again, are in black here, are smaller on average. And you can kind of see that there is no bump in the single connections. I think that's because single connections are going to be less likely. Less likely to have learned and become in the strong state. Although there's probably a mixture of stuff in here because some of those single connections are actually dual connections that they didn't find. But the dual connections have this bump that looks like a large that they have larger synapses, more likely to have larger synapses. And then in H, what they do is they use this model and they say, if we were to look at the synapses that have one connection, two connection, and now they look at the three connection synapses, does the fraction of estimated large synapses go up? And that's what they find. They find that that as you have larger number of synapses between these two cells from A to B, the likelihood that they're large synapses goes up. I think this is just to step back for a second. This shows how relatively straightforward it is to get stuff out of connectomes, because the strength of a synapse, as really determined by the number of, these are all excitatory synapses, really determined by the number of glutamate receptors. That correlates very well with the size of the postsynaptic density, which they also measure in this paper. That correlates very well, both of them, with the volume of the dendritic spine, which is the main thing that they use in this paper. And it also correlates with the just rough number of synapses between things. And if you look at papers like the fruit fly papers, the number of synapses is often used between two cells, is often used as a proxy for the strength of functional connection between cells. So this is just showing that in cortex. So their last key figure here is really a replotting of this as kind of a grand summary. They take all of the dual synapses and they plot each of those as a point in this space. So, for example, this point up here is going to, one of the synapses is going to have a volume of, let's say, 10 to the minus 2 cubic microns, and the other one is going to have a volume of 10 to the minus 1 cubic microns. That's why they put that dot there. They mirror all of these because we could have chosen each synapse as synapse 1 or synapse 2. And they're plotting all of those synapse pairs in this space. And so what would you expect in an image like this? Well, first of all, if they were correlated in size, you would expect them to lie along the line, the 45-degree line here. And whereas they are correlated in size, and they do lie along that line, there is definitely more structure than just correlation between these. There is a hotspot here in the small range and a hotspot here in the large range, and they're quantifying that with this heat map that is coming from fitting their binary model. the joint probabilities that they are getting from that fitting are shown here. So they're saying for these synapse pairs, the same pre and postsynaptic, there is 0.49% probability that they're both small, 0.33 probability that they're both large, and much less probability that they're a mixture of large and small on the diagonals. This is just showing the this without that heat map. So, you know, even without these fits and stuff, you can still see that there is definitely two hotspots there. So one thing they do in a supplement is they separate this data based upon whether the two synapses are closer together than the mean of 46.5 microns apart, or if they're larger. There's more separated than that. Let me read this to make sure I got that right. Dual connections of in A, dual connections of synapse pairs less than 46.5 microns apart, and in B, dual connections that are more than 46.5 microns apart. And the fact that you can see the same picture, the same correlation between these is it is incredibly it's incredibly telling. It's saying that this correlation between these sizes and these small and large variables is not due to being on the same dendritic branch, right? Because the ones that are far apart are almost certainly not on the same dendritic branch. They're not on the same little part of the dendritic branch. So this is evidence that you can model whole cells from a learning point of view as a single cell as opposed to branches. It may not be the strongest evidence. I know we've covered papers that that argue against that. I'm sure there is some argument to be made that different branches of dendrites, different dendritic branches, have slightly different spiking that could lead to different learning. But this is making an argument that, hey, the correlations of the learning between synapses really is not dependent that much on how far or how close those synapses are. It's a key finding. The other thing that they do here is they plot the dual connection correlations in A and D, and then they plot them for. Let me read how they do this here. So B is a subset of the data for axon preserved shuffling. So this is saying that A is the data. A is the data that you've already seen. D, E, and F are going to be different shuffles. D is just a shuffle of synapse pairs, but E is a shuffle of the axon preserved pairs. And so what that means, they have a better figure here. I'm going to jump to it. Oh, they don't have a better figure here. Is it in a supplement? I think it was in the supplement. Oh, darn. It was in the supplement. In any case, it's essentially saying I'm going to make sure that I am swapping, I'm shuffling synapses, but I'm going to make sure that it's preserving the same axon. That's what's shown in E. Or I'm going to shuffle them, making sure that I'm preserving the same dendrite. That's what's shown in F. And the key thing is that if you do any shuffling, you lose the correlation, what does that mean? It means that this is not a strengthening that is based upon the axon itself. It's not saying all of the learning was presynaptic for this axon. This axon really had, or this presynaptic cell was really strengthening all its synapses, or this postsynaptic really strengthening all its synapses. It really is correlations between synapses that have the same pre and postsynaptic cell, just like you would think in classic Hebbian plasticity. So I'm going to go through the conclusions. I want to give a pause here for any questions.
I didn't have a specific question. It's just that it was occurring to me that, yeah, so there is a lot of room for what we might consider analog types of coding when you think about, okay, synapses could potentially be of any size. And the same way, there's a lot of room for complicated analog computations when you think about the things you can do when modeling branches specifically and modeling in great detail how the different branches of neurons are calculating, computing, etc. But then you see this kind of what I consider constraints being imposed, some sort of mechanistic or learning-induced constraint, pushing everything in one direction or another and simplifying. So you've got both this bimodal aspect and what you were mentioning about you could definitely model this as a single cell. I just find it very interesting to consider where that is super useful when you're thinking about what does a piece of brain have to accomplish versus where is it not. So then when I think about prefrontal cortex and the types of operations that are taking place there, it does seem useful to have all these constraints. But then if you have some other system, maybe a sensory system, something like auditory perception, where you're dealing with analog signals, it seems like you might then make use of more of the rest of the sort of computational room that's there when you're not constraining yourself as much. I know this is kind of a little bit off to the side, but it's just what's been going through my head as I was listening to this.
And they stress in this paper multiple times that this is something that they're seeing between layer 2-3 pyramidal cells in visual cortex. And basically this type of analysis would have to be done for every pair of cell types to really to see if that pair of cell types their synapses behave this way. So it's putting constraints, but it's only putting constraints on this particular visual system and this particular set of layer 2-3 cells. So, okay, so let me, unless there's any other questions, we'll have discussion pretty soon here. I'm going to go through what they have for their conclusions. So first of all, they just summarize this. Our synapse size correlations are specific to pairs of synapses that show both the same presynaptic and postsynaptic layer 2-3 pyramidal cells, similar to these other findings. And they point out that these other findings are they simply have much smaller numbers than that they could not do this analysis. We have further demonstrated that the correlations exist even for large spatial separations between synapses. I think this is a real key thing in this paper that none of these other papers could do because they were all on very small volumes, except this MATA paper, which we should cover at some point. I'll talk a little bit about it in a second. More importantly, we have shown that the correlations are confined to the binary latent variables in our synapse size model. The log normal analog variables exhibit little or no correlation. Okay, so the only correlation that they're seeing is in a binary state. It's either in small or large, and everything else is noise. And what do they speculate about this noise? They say the correlations in the binary variables could arise from a Hebbian or other synaptic plasticity rule driven by pre and postsynaptic activity. that are relatively uniform across neuronal arbors. That's like I was saying, this is it's a whole cell spiking that is really being presumably involving the learning process. We speculate that much of the analog variation, because there's a lot of analog variation here, right? You could think, my God, why would there be such large differences in sizes of these synapses if what they really need is just a small and a large? And they say, we speculate that much of the analog variation arises from spontaneous dynamical fluctuations that have been observed at single dendritic spines through time-lapse imaging. So other papers, other labs have done time-lapse imaging of fluorescently labeled dendrites and seen that the sizes of the synapses grow and shrink all the time. They may stay on average large, or they may stay on average small, but there is a lot of dynamic fluctuation in there, and that just might be as good as the biology can constrain the size. So the models that they suggest, okay, and this gets to like how learning occurs. Our latent variable model is consistent with the scenario in which synapses have binary switches that are flipped by activity-dependent plasticity. Switch-like behavior could arise from bistable networks of molecular interactions at synapses. They quote Lissman, 1985. I believe this is one of the beginning papers that was talking about CAM kinase 2 and the autophosphorylation of CAM kinase 2 as the possible molecular switch that gets turned on in a Hebbian plasticity event. So the idea is that if the pre- and postsynaptic cells fire together, there will be a calcium input to the dendritic spine. That calcium input will activate kinases that will phosphorylate. I hope I'm getting this right. Phosphorylate the molecule CAM kinase. And it has, once it's phosphorylated, it has the ability to phosphorylate itself. And so the population of CAM kinase molecules, CAM kinase 2 molecules, will keep themselves in that on state. And that is one of the key things that is put forward as a possible learning mechanism. And this is evidence in its favor. They say in a second scenario, synapses are intrinsically, as opposed to that, synapses could be intrinsically volatile, and by modality might arise from learning driving them to the extremes. There might be a size that's, you can get a synapse to a large size, but it can't go any further. And you can get a synapse to a small size, but it can't go any further without essentially dying off. And they argue against that because I think their data is very strongly arguing against that. We'd like to suggest that the first scenario of binary switches is somewhat more plausible for two reasons. First, it is unclear how the second scenario could lead to strong correlations in the binary variable. If you're just shoving all the synapses to the top or to the to their extremes, it's not clear that you would get a correlation with a binary variable. Second, it's unclear how the second scenario could be consistent with little or no correlation that remains in our data once the contribution of that binary latent variable is removed. So they argue that this is, more research needs to be done, essentially.
That second point specifically, that's the one that gets me.
Okay.
Because when you take that out, then you would definitely expect to still see a correlation if it was a learning effect. If it was just an activity-driven effect.
let me throw that to here, and then I'd like you to unpack that a little bit more. This paper, this Mata paper that they also reference here, they have a completely different model of synapses and they do model it as if you have LTP, it would drive it to the extreme. And it would either drive both synapses to being big or drive both synapses to being small. And I think this is kind of the second scenario that they're arguing against.
But that's exactly why I said it was the second part, when they said the second argument, if you take out that first element, if you remove that, then you should still see a correlation if this was an effect where it was being driven to an extreme rather than a switch. Because it, I mean, unless they just got unlucky and they only found cells where it had already been driven to an extreme and they didn't find anything anywhere in between.
Yeah. Yeah, absolutely. The only other thing I want to point out is that this Bartol et al. Paper that was, I think, the first one to really use this type of analysis. They had a very small number by today's standards of this was in hippocampus CA1. Okay, so they were looking at pairs of synapses in hippocampus CA1 with the same pre and postsynaptic cell. Very small region, so all these synapses were very close to each other, and they saw an amazingly good correlation between these pairs of synapses, although the way they treated this triple synapse is kind of dicey. But from this amazingly good correlation, they argued that you could you must have higher bits of precision. So they were actually arguing that you had multiple bits of precision for each synapse. Clearly, that is that analysis is at extreme odds with the paper that we just went through, and I think either the hippocampus is different, which is totally possible. So it'd be lovely to see this type of analysis in a hippocampus again. I haven't seen it, but if anybody knows it, please tell me.
Can you remind me if this, when they said multiple bits, did they mean in terms of there being, say, if we were to say map this onto the paper we're talking about today, multiple modal regions, or just that it's multiple bits when you take into account that there are two modal regions and then you have more and more synapses, so that you get more bits that way.
My recollection of the paper is they were arguing this strictly from how correlated the two are over the range of the synapses size. And they were saying, hey, if we have kind of a spread over this full range, and they're so well correlated over this full range, then there has to be a mechanism that keeps them locked together in that range. So if there was if they were perfectly correlated, but they were all either small or large, that would be completely compatible with this paper. But what they were arguing is that we have a range of sizes and they're perfectly correlated. And from that, again, I don't know if I don't know how good the analysis is here, but they argued that if you wanted to maintain that amount, then the individual synapses would have to keep track of their learning history with this many bits of precision. And that's how they argued that there were 26 distinguishable intervals of spine sizes.
And there was also evidence of a bimodal distribution of connection strengths from electrophysiology, I believe. I think they mentioned some of it in this paper. They've referenced a Song et al. Paper, but there was also work that Markram did all the way back in like 1997 on paired recordings in neocortex.
Oh, really?
So, yeah, we've seen there have been some of these bimodal findings before, and from EFIS.
Yeah, so I know they referenced a bunch of papers that were kind of like, people have argued this before and people have argued this against this before. But this is a pretty strong, they have so many synapse pairs to make this argument. It seems like that was the key thing, that it's a good example of volume connectomics and dense tracing being what you need to answer these key neuroscience questions. Yeah. So the floor is open. Ariel?
I think I saw that the preprint for this paper was put up in 2019 initially, which means it's been a few years since they initially published this. Are you aware of whether there's been any follow-up or whether other groups have replicated, extended, changed, or anything? Or is this still like the best data we have or analysis we have on this question?
I am not aware of it. I have been to conferences where Dorkenwald has presented this, and I have not seen him present new data. But I would be surprised if somebody is not doing it, because there's a bunch of data sets now that could support even larger statistics on this. We really should go through the Mata paper at some point, because that talks about different synapse, different cell types, and seeing Hebbian plasticity signature in some, but not in others. So I'm curious. I mean, are people convinced by this paper? Or are they or are they not? Yes, go ahead.
Kunal, I think your microphone is, yeah, there we go.
Yeah, hey. No, I think I was actually just thinking along the lines of what you just asked. I was thinking, so they're looking at a few hundred neurons and a few thousand synapses. And you took a log function and you found this double normal distribution. I mean, I would imagine with this data size, again, obviously I've not tried it, but you could come up with another function that might come up with, a different distribution. Are we giving too much importance or getting, based on a log function, right? A lot of this analysis is based on that one function. And I was just thinking,
this is exactly
an assumption.
This is exactly the kind of argument that I'm looking for. So let me play the defender on this, but I want to hear the arguments against it. So when I look at that, actually, let me share my screen again. So when I look at this, okay, so the orange spots are, I guess you can say those orange spots are from their model that they're fitting, right? So they're fitting a model that has a binary latent variable and a log normal noise on top of that. So of course it's going to look like two bumps. But if we look at this this on the left here, I can still see two bumps. And if it was a different model, I'm more convinced by looking at that and saying, wow, these are well correlated and they're falling into two bumps.
So you're convinced it would still look like two bumps if you don't plot it log normally, but instead plot it, say, along some other...
If you don't plot it log normally, everything will be squashed, I guess, which is a reasonable statement. And I think there's probably arguments for things being log normal because, this might prove fraught if I make this statement, but isn't the idea that so you have a central limit theorem for normality? So if you're adding a bunch of random variables together, then the mean of adding samples from random variables together, you'll get a normal distribution. But if you have a process where you have a bunch of things coming in and they're multiplying with each other, then you would get a log normal distribution. Somebody help me that has a better statistics background. Ariel?
Yeah, I mean, I can hear that you're quoting central limit theory, but I don't remember it that well. Maybe Ariel does.
I remember the first part. I don't remember the bit about multiplication and how you get log normals.
Okay. In any case, that's my I guess let me throw the question back at the asker. If there was a different model, what do you think might be the most likely one to check next?
I mean, we're thinking math here versus biology, right? I would almost think from, I mean, why have a function? I'm not sure if that's how biology works, is what I would initially think. It's just the raw data that you have. Because, like I said, I've not tried. This kind of data size, I'm sure we could come up with another function that'll show us patterns. And again, I think I do see the merit in it, and in some sense, this does make sense to me. You're taking the log, like you said, you're basically taking the multiplier, which does make sense to me. But that is probably the little doubt that comes in my mind. I think this is credible work. But that was just one thing that was coming in my mind, with this paper.
The other thing that again appeals to me about this, and because I like simple models that kind of make sense mechanically, is I can imagine having a molecular bistable switch like CAM kinase 2 or something else that flips over. I was actually having a conversation, and I can imagine that being activated in a learning moment with calcium. I can imagine it being stable over years potentially, right? Because that is a binary lock that has been shoved to one side, and it can't unphosphorylate itself. But coming up with mechanisms that can support a analog or a very fine-grained number of stable states for these little tiny synapses that are far smaller than a cubic micron, right? Coming up with something like that is harder to imagine, and there aren't models that I've seen that really do that well. I haven't seen models that say this is something that can withstand the buffeting of months and years of protein turnover and retain a memory. And so this retaining of a memory by basing it on a binary switch at every synapse is very appealing to me. Ariel?
I guess one thing I would find relevant to this is I actually don't know what the size of the fluctuations in like spine size or synapse size are for synapses that they don't think have been necessarily strengthened into these large sizes, or that have been. I guess one thing that would be interesting is if you could track in a similar way, like a whole bunch of synapses like this paper did, but somehow you were able to do with like live imaging and fluorescence and just see how much like bigger and smaller they get, and then be able to compare it to like I guess like the fluctuations within a synapse to the like distributions between them, because I wonder if the like variation in individual synapse size is much smaller or much larger, such that you could expect it to be, influencing what you're seeing. Or if you had like really small variation in like the size of synapses over time, then that would support the idea that if you see like really big differences in how big they are, it's just due to these changes.
That's a good point, but that's where, for instance, when I see additional support coming from EFIS studies showing also a bimodal distribution in PSP strengths or connectivity by doing these very careful recordings, that helps me a bit. At least it makes me a little bit more confident.
Yeah, I mean, I'm not super skeptical of this idea. I just wanted that like the good, strong confirming evidence. But sorry, Ken, you go.
Oh, well, I was going to say they in that paragraph when they're talking about these variations, they say that they have done time-lapse imaging in other papers. They say experiments have shown large dynamical fluctuations persist even after activity is pharmacologically blocked. And they give a list of 4 papers. I didn't look up any of them, but there does seem to be papers that probably do quantify volumes changing over time. Yeah. Randall, I wanted to follow up. Do you happen to know how? I'd love to take a look at that paper that you're talking about with the EFIS. If it's in this, I still don't know where it is, so shoot me an email.
No, the Markram paper wasn't in there. That's elsewhere. It's an older paper, but it's one I just happened to remember.
Do you remember if they had a lot of statistics, or if it was kind of like?
I'll have to check. I know I can find it, so I'll find it for you. I'll send it to you.
Cool. All right, we're coming up on the hour. Any other stuff? Like I said, I butchered the presentation of this paper, I'm sure, but I love this paper. I've been looking at it. I've been wanting to do it for a journal club for a while, and I feel like this is definitely something that should be nominated for one of the Aspirational Neuroscience Awards. I don't know if it should win, but it's definitely hits
it's
all
impact,
of
right? Yeah, the impact is really important if it's true. Yeah. And the other thing is just that this makes a mockery of the 16 hours I spent just last week improving very carefully the model of PSPs that I'm using.
Yeah. Yes.
Just want to flag on the note of nominations that we're going to have a discussion. The next journal club will not actually be a journal club. It will be a papers discussion nomination for the Aspirational Neuroscience Awards. So if any of you are listening here, if you have any papers in mind you think should get an award or have advanced memory decoding, neuroscience sort of stuff over the last few years or so, you should nominate them. You can either nominate them on the website, just submit a form. You can just email me about it, or you can come along to the discussion group in 2 weeks from now, where we will be talking about these different papers and the papers we've discussed and papers we haven't discussed and everything associated with this, trying to narrow down some candidates for who's done good work and should get an award. So please come then.
Great.
And so just to be clear, right, this event meeting in 2 weeks, this is just as open to everyone as these meetings are, right? It's
correct.
Yeah, and will be advertised the same.
Yes. Well, I'm going to advertise it the same way. Yeah.
Okay. Awesome. Thanks, everybody.
Yeah.
Thank you, Ken.
So it's simple, Ken. It's binary quantized states in the brain.
This was great, shocking,
yeah.
And yeah, that's something I really want to know if it's true now, because then that's something to keep in mind, at least for pyramidal neurons in certain cortical areas, and maybe hippocampal pyramidal neurons as well. Yeah.
It'd also be nice to see like a follow-up study with expansion microscopy that might look for the molecular mechanism underneath these, because you could imagine having, 1 turn red and one turn green.
Indeed. Yeah. That'd be fantastic. Okay.
Okay.
So maybe you could launch a prize to have some motivation out there for someone to do that study.
I think there's probably plenty of motivation already, but it'd be great if I run into somebody that can do it. I think I know who
Okay.
can. I will suggest this.
Perfect. Thanks again, everyone.
Thanks.
Thanks, Ken. Bye all.